Hello, everyone, and welcome to our seminar in our virtual seminar series on pluripotent stem cells. My name is Amanda. I work in scientific marketing here at STEMCELL Technologies, and I'll be moderating the seminar and question period today. Today's seminar is titled Quality Control Guidelines for Clinical Grade Human-Induced Pluripotent Stem Cell Lines, presented by Drs. Stephen Sullivan and Andrew Gaffney. The first portion of this seminar has been pre-recorded. Following the presentation, there will be a live Q&A session with Andrew. Dr. Sullivan is a program manager at Global Alliance for iPSC Therapies, otherwise known as GATE. And since 2017, Dr. Sullivan has overseen operations at GATE, supporting implementation and clinical application of therapies derived from pluripotent stem cells for the benefit of patients globally. Dr. Andrew Gaffney completed his PhD in pediatric oncology from the University of Leeds, UK, where he successfully established a human ESL disease model of Ewing sarcoma. Currently, Andrew works closely with renowned stem cell scientists in both industry and academia to monitor the requirements needed for establishing successful HPSC-based disease models, drug screens, and cell therapy applications as part of stem cell strategic alliances. Before we start, as a reminder to the audience, if you'd like to ask a question during the webinar, please use the Q&A panel or chat box on the right side of your screen, and we will address those questions at the end of the presentation. And with that, I'm going to pass it over to Stephen and Andrew to start the presentation.
Okay, thanks Amanda for the introduction, and I'm very happy to be here today and joined by Stephen from the Global Alliance for iPS Cell Therapies to discuss the guidelines, definitions, and recommendations for the production of clinical grade iPS cells. To briefly run through the agenda for this webinar, we'll be starting with a lesson from history on the importance of cell characterization, and then move on to the specifics of iPS cell quality, where I'll spend some time defining terms and the quality attributes between the different grades of cell lines. And all of this feeds into the end goal, which is, of course, treating patients safely. Following this, Stephen will be discussing the GATE initiative, including information on the expertise required to produce safe therapeutics from iPS cells, explaining why it's important to build a holistic vision for translation, and the risks and mitigation strategies involved in creating a cell therapy. We'll then touch upon the current clinical trials ongoing using iPS cells and discuss the future for these therapies. So to begin, I'd like to focus on an example of why it's important to learn from those who have gone before us. And in this instance, it's the field of MSCs. So there have been many contradictory findings about what some people call mesenchymal stem cells, or MSCs, which have included their origin, developmental potential, their biological functions, and also their therapeutic uses. And because of this, clinicians and societies have recommended that this term, the mesenchymal stem cell, should actually be abandoned. Now, the original MSC concept described the isolation of a type of stem cell found in bone marrow. And this work showed that certain cell populations in the supportive tissue of the bone marrow could differentiate into bone, cartilage, and fat, and provide some of the signals needed for hematopoietic stem cells to give rise to blood cell types. So over the next 10 years, MSCs went on to be reported in many other tissues, and different labs are even using different panels of cell surface markers to characterize MSCs. So the International Society for Cell Therapy, ISCT, stepped in to recommend a new name for these cells, which was multipotent mesenchymal stromal cells. But the original mesenchymal stem cell name actually seems to have stuck. So according to the literature, MSCs are most often isolated from bone marrow and adipose tissue, or fat. They've also been identified in perinatal tissues, that's the umbilical cord, Wharton's jelly, the amnion, and the placenta, as well as other sources, including baby teeth and menstrual blood. Some groups report that MSCs are most common in fat. Others state that they are associated with blood vessels throughout the body's connective tissues. Some say that MSCs are exceedingly rare, while others say that they're very abundant. So as you can see, if you don't define something at the get-go, you end up with a lot of confusion in the long run. Now, we know that there's approximately 1,000 clinical trials worldwide that have used MSCs, but as you can see from the chart on the left, the numbers of trials were rising steadily until about 2018, where we actually saw a dramatic drop. And the authors of this paper suggest that this could be because there's been limited success in achieving outcome measures for efficacy. Now, because of the lack of standardization in this field, we know that there are distinct differences in the MSCs that are being generated in different locations. And this has made it tremendously difficult to perform meta-analyses of the clinical trial data. And this is a great example to demonstrate that agreeing upon standards by consensus is easier the earlier it is done, because people become committed to the protocol that they're using. But I wanted to include this quote from Douglas Sipp, Pamela Roby, and Lee Turner, where they state that whatever the ultimate identity and biological activity of the cells formerly known as MSCs, the scientific principles that guide their clinical development and use must be the same as those for other new therapies. Precision, validation, characterization, objectivity, and the abandonment of terms and conceptual models that mislead more than they illuminate. And I think that this is also a great message to move forward with when discussing other therapeutic cell types. So how do we solve the issues that we've discussed so far? Well, projects like the Human Cell Atlas at the Broad Institute are looking to create comprehensive reference maps of all human cells, and this could shed light on the components of different tissues, which could aid the MSC field tremendously. But we now know that a focused undertaking by scientists is going to be required for long-term success, and groups such as the International Stem Cell Forum are going to be very important, and this is a consortium that was made to characterize human pluripotent stem cells. Here you can see that one of the groups that spun out of the forum was the International Stem Cell Initiative. And this has been a tremendous global effort since the birth of human embryonic stem cells to characterize, firstly, ES cell-associated surface markers, secondly, to assess defined feeder-free culture systems for ES cells, and finally, and more recently, to quantify techniques to assess the pluripotency of ES and iPS cells. As well as ISCI, the International Stem Cell Banking Initiative, or ISCBI, was also formed to assist in the development of an agreed set of international standards for banking, characterization, and testing. And finally, the Ethics Working Party, or EWP, was formed to define transparent ethical frameworks for working with these cells. Now, for anyone who's interested in learning more about the pluripotent stem cell quality standards and banking standards defined by these organizations, I'd highly recommend you view this series of short seminars and panel discussions that were recorded as part of the Stem Cell Technologies and Nature Research Roundtable. This includes discussions from Peter Andrews, an original founder of the International Stem Cell Initiative, and Glenn Stacey, the original founder of the ISCBI. You can find these discussions at stemcell.com/Nature Research Roundtable HPSC Quality. Okay, so next I'd like to move on to the topic of specifying the quality of iPS cells specifically, and we're going to be discussing this in 3 parts, including the GMP manufacturing process and the differences between research grade and clinical grade iPS cells. Now, before we go into this section, I think it's important to clarify some terms that we're going to be using that have often had multiple different meanings in the stem cell community. So we've set out our definitions here. And the first term is research grade, which refers to products that are intended for use in research. Now, this is very difficult to a clinical grade product, which are produced with the intention of ultimately being used in clinical applications. We're also going to be referring to ancillary materials or raw materials, which is the term used in Europe, which are components that are used in the manufacturing of therapeutics that are not intended to be part of the final cell therapy product. And finally, the term GMP, which is not a grade, but is the manufacturing process that's required to conform to guidelines recommended by regulatory bodies to ensure the consistent production of a product from batch to batch. I'll now briefly hand you over to Stephen to explain why someone might need to work under GMP.
Before we reference GMP and consistency, I just wanted to say a few words about scalable manufacturing and the importance of variability and identifying variability in that process. So obviously with the cell, you're going to have biological variability. The cell is going to have some innate variability because of its nature. And this unfortunately is unavoidable. We have to engineer around these behaviors, if you will, that are beyond the control of the manufacturer. But there are lots of other types of variability that contribute to you know, a manufacturing process going awry, and these all require careful understanding and management. So things like process development, process variability, analytical variability, and other forms of variability. So the key thing when you're focusing on GMP, really what you're looking at is you want a consistent result at the end of the day, and through GMP, through that practice, you get a consistent result. But the term GMP in and of itself doesn't inherently tell you to what specification that is done. So the key thing is, if you see something labeled GMP, it's important to know the specifications to which the GMP process has been done. The devil is in the detail, so to speak. Back to you there, Andrew.
Okay, thanks, Stephen. So to illustrate what's involved in creating a GMP-ready process, I wanted to start with a workflow that I'm sure everyone on this call is highly familiar with, and that's this traditional iPSC workflow. As I'm sure, as we all look to do, we can go through it and look to optimize the process as much as possible. So we start with acquiring some starting tissue, which in most cases involves taking a punch biopsy of the skin. Then we expand these cells out to make sure we're starting with a large enough population. We reprogram the fibroblasts using a method that's as efficient as possible and is going to give us a good number of reprogrammed clones. And ideally, we'd use a vector that is also non-integrating. Once we've established the iPS clones in culture, we want to maintain and expand these cells in a culture system that ideally is defined and feeder-free. And of course, we want to bank these cells, creating both master cell banks and working cell banks. And we also want to use a method that's going to give us good recovery rates and viability after we've thawed the cells. And finally, we want to perform a characterization step where we assay for particular quality traits of pluripotent stem cells to make sure the cells are what we think they are. But if we want to transition this workflow into a manufacturing process that's GMP ready, here you can see that there's a tremendous number of additional controls that need to be implemented in every single aspect of that workflow to make sure that the product you're manufacturing is consistent. But even if you do implement things like non-integrating reprogramming vectors and xeno-free cell culture media, for example, that's not what constitutes a GMP-ready manufacturing process. This is really all about traceability and documentation. So in order to achieve this, strict batch records and standard operating procedures need to be prepared. There's big differences between a protocol and an SOP, which is a version control document, and you should be able to refer back to a particular version for any manufacturing run. It's important that the GMP production team are trained and qualified to perform particular processes. The GMP facility has to be prepared, which includes managing work environments and certifying equipment. Characterization assays have to be qualified, QC documents have to be prepared, and viral testing has to be performed on master cell banks. Now, this might seem very detailed, but to further demonstrate what's involved, I'll highlight just one of these controls, and that's the supply chain. Now, within this section, you're required to audit your suppliers, execute supply agreements, qualify your ancillary materials, and confirm certificate of analysis results for 3 different lots. Your cold storage needs to be qualified, incoming ancillary materials need to be inspected, and you also need to control the flow of these materials as well. Now, this is just a selection of what's involved in the supply chain, and this level of detail is also present over the entire manufacturing process, making this an extremely detailed operation. Now, once you've completed this GMP manufacturing run, you'll have produced a master cell bank or a seed stock. The cells will have passed defined release criteria as well as QC assays, and they'll have undergone viral testing. Everything will have been banked under GMP, traceability criteria will have been met, and the storage procedures will also have been documented. But these master cell banks can also be used to create research-grade working cell banks. And these matched cells can actually be put to use by research labs to perform preclinical studies or to optimize things like differentiation protocols. And this will allow researchers to focus on creating clinically relevant cell types, such as dopaminergic neurons or cardiomyocytes, for example. Now, as I mentioned earlier, an ancillary material or raw material is a component that is used for manufacturing therapeutics that's not intended to be part of the final product. I should state that technically there are no GMPs for ancillary materials, so it's important for clinical trial sponsors to do their due diligence in qualifying their use for specific applications. Now, this table shown here is a risk level taken from USP Chapter 1043, and this gives examples of reagents that you might be using to manufacture your cells. And if we take MTESA1 as an example, which is a PSC maintenance media that we manufacture here at STEMCELL Technologies, you can see that this would fall under the example of a tissue culture media. And then you can then see on the right the suggested qualifications, such as confirming certificate of analysis results, cross-referencing a drug master file, which MTESA1 has, as well as suggesting you should use a CGMP manufactured media, which MTESA1 is. At STEMCELL, we can help support your qualification activities with things such as providing C of A's, C of O's, risk statements, provide you with quality questionnaires, and we also welcome audits of our manufacturing facilities here in Vancouver, in Canada. I also just want to know that this USP is actually a US publication. However, other global jurisdictions follow similar risk-based approaches, so a lot of this detail will actually be relevant everywhere in the world. Okay, so when choosing an ancillary material, it's very important that you examine the supplier's claims and definitions about the products and make sure that they're sufficient for your particular needs. There's actually no industry standards for definitions of the different terms. And so from supplier to supplier, you might find that you're being given the same definition, but with different meanings. So let's take the animal origin free or AOF classification, which at STEMCELL we define as not containing any ingredients derived from animals, and that includes humans. As part of our definition, we also state that the components used to manufacture these raw material ingredients are also not derived from an animal. So our AOF term is true to the secondary level of manufacturing. Now, if we compare this to another supplier's definition, you can see that they state that their product is completely animal origin free medium, but at the primary level of manufacturing. Now, the implications here of there being animal components in the media that you may not have realized could mean that the regulatory authorities may come back at a later date and require additional testing to be performed on reagents or the final cell product, such as adventitious agent testing, for example. So it's good to have this clarity up front so you know what you might need to expect in the long run. For anyone who's interested in learning more about our other cell culture media definitions, such as xeno-free, serum-free, or animal component-free, you can visit stemcell.com/how-do-we-define-our-media. Now, as I mentioned earlier, when we're referring to the term GMP, we're actually referring to a process, whereas research grade and clinical grade is a set of specifications for the intended use of a product. And in this case, we're referring to the product as being iPSC lines. At STEMCELL Technologies, we frequently receive inquiries from researchers asking what the appropriate assays are to monitor for pluripotent stem cell quality. And in truth, the answer to that is that there's no strict enforcements for research grade, just recommendations. And of course, in the research lab, we have to be considerate about the costs and the time involved in each individual assay. And we shouldn't expect researchers to be sacrificing their advancement of science by pouring time and money into over-the-top quality testing, which could end up being overkill for your specific application. So we generally find that the quality tests described on the screen here are quite commonplace, and that includes checking the morphology of your iPS cells, which can be done frequently under the microscope. And you're looking for that round, tightly packed cell morphology with a large nucleus to cytoplasm ratio. Genetic stability, where ideally you'd perform a G-band along with an array-based method such as a SNP array. Although in truth, if you're already routinely karyotyping, that would probably already put you towards the top of the pack when it comes to monitoring the health of these cells. Analyzing undifferentiated marker expression by flow cytometry, that should be routine practice. as well as demonstrating the potential for trilineage differentiation of your cells by either the teratoma assay or through spontaneous or directed in vitro differentiation. Now, here's the results of an interesting study that was performed by iCell, where they identified that 34% of all lines submitted to them for quality analysis failed their minimum standards. So there were over 800 lines analyzed in this project. And of the ones that failed, you can see that an overwhelming number did so because of an abnormal carrier type. That's 61% of them. Another 25% failed due to viability issues, which meant that the cells basically couldn't be grown once they were received by iCell. 10% due to identity, indicating that these lines were either mismatched or mixed populations. And 4% failed due to sterility issues. Now, it should be noted that these lines were submitted to iCell for distribution to the wider scientific community and were believed to be of good quality by the investigators. So we should just take a minute to stop and think that if iCell failed 34% of these lines, how many lines are there out there in the current PSC research community that are not up to standard? And so I'd really encourage everyone to monitor the quality attributes of your cell lines as frequently as possible, but for your particular situation. And with that, I'd like to hand the presentation over to Stephen, who will begin by taking you through the quality aspects of clinical grade iPS cells.
So as Andrew said, you choose the specification in which you undertake a process to fit the end use of the cells and their derivatives. So to begin with, what I thought was I'd share two papers with you, which are really good kind of primer articles for considering what is required when banking iPS cells or the scalable manufacture of iPS cells, where it's therapeutic development that you're considering. So you're considering using these cells as a starting material for clinical use. So, and in Gates' instance, we're developing a network of haplobanks that will house starting material for allogeneic iPS cell therapeutics. So basically, the iPS cells that are to be housed in the Gate network will be specifically generated to a specification where they can be used successfully as appropriate starting material for allogeneic therapeutics, off-the-shelf therapeutics, if you will. So the paper on the right is generated by the ISCBI, the International Stem Cell Banking Initiative, which covers requirements from a clinical banking perspective. And then the paper listed on the right-hand side is focused on what are the requirements from a manufacturing point of view. And this was published by the UK Regenerative Medicine Platform. So that they would be the first two areas that I would two articles I would consider reading when considering making allogeneic iPS cell therapeutics. So we talked a lot about the importance of standards. One of the key elements to standards as well is they're only as powerful as their use. So for example, if I have a standard and Andrew has another standard, then our capacity to work quickly together will be diminished. Whereas if we both have the same standard and understand the same thing by the same term, then we'd be able to work a lot better, more efficiently together. So with that kind of in mind, when Gate decided it was going to initiate this global network, it decided that actually we need to have a series of quality workshops where we really discuss what we mean by iPS cell quality in terms that relate to its use as a starting material for allogeneic iPS cell therapeutics. So this is just a list of some of the organizations that attended our quality workshops on iPS cell quality, clinical grade iPS cell quality. And This next slide shows the publication that we released back in 2018, which basically highlights in detail the QC guidelines that were agreed at these quality workshops. And the key thing is that, you know, as I said before, standards are only useful if people adopt and use them, and that they're common between different stakeholders. So a lot of discussion, a lot of reconciliation and development of these guidelines were undertaken. And now we've published these consensus guidelines, which are the first iteration of what we at GATE would classify a clinical grade iPS cell line to be housed in our global networks. Now, technically, you do need a therapeutic to be on the market and to have got regulatory approval before an iPS cell is really, considered clinical grade. But in the absence of directive regulatory advice, the community has to align around, I suppose, the best guesses of what will be required by the regulators later on. So really, you know, as Andrew was talking about earlier on, you know, that the community, if it waits for regulators to direct what is acceptable or what is not, without any intrinsic alignment, an awful lot of work will not be fit for purpose. And there'll be a lot of confusion, as we've seen with other cell types. So our goal with this first iteration of the QC guidelines for these clinical grade iPS cells is to facilitate collaboration between members of the network and accelerate therapeutic development. And ultimately, what we hope through such initiatives such as this is that there'll be a greater clinical impact at the end of the day. So more patients will be immuno-matched, for example, in the immunobanks, and the quality standards will be consistent between all networks, which would then allow a degree of accelerated translation. And just to say that this is the first iteration of the GATE QC guidelines. Obviously, we'll be coming back to these regularly and adjusting them so they're fit for purpose going forward. So, you know, when I was considering, when I was considering this webinar, I was considering that, you know, really probably at the next GATE meeting where we discuss these QC guidelines, obviously one of the things that we'll be considering is, should we add COVID-19 testing, for example, which would seem to be an appropriate process for me, because obviously if you generated a therapeutic from a clinical grade iPS cell line that turned out to have COVID-19, then you'd have a huge negative impact for patients later on. So that's just highlighting how the more we know and the more technology evolves, the more we have to update our quality control guidelines so that really the material is fit for purpose for its end use. Standards need to be feasible, acceptable, and suitable so that they're initially adopted, and that their benefits around accelerated translation are realized. But we have to realize that, standards need to be developed by the community in order to be accepted by the community. And engagement and having these quality workshops that are open and all stakeholders can come and describe their particular recommendations and then hammer out a consensus from there. That's very important. One of the elements that maybe we don't consider every day if we're proof of concept basic researchers would be how the absence of standardization drives up costs. So for example, here I've some of the major risks that face iPS cells as they go towards the clinic. And one of the elements is that the therapies would not be commercially viable. So if you imagine, everybody is doing their own thing to a degree, you lose a lot of economies of scale, and the costs remain prohibitively high in terms of developing your therapy. Low adaptation of different technologies. So if everyone's using a slightly different technology, then again feeds into a big risk in terms of translating the therapeutic development efficiently, or generating therapy efficiently. And also, if we don't have a discussion around appropriate regulatory requirements, so that researchers and developers have an open discussion with experts in regulatory affairs, then what you're going to see is different countries ending up with very different regulatory requirements, asking for different tests and so forth. And this also drives up the cost unnecessarily without any real benefit to the patient, no reduction of relative danger or risk to the patient. So really, I think the key thing with the standardization of the field as a whole is it gives us clarity, it reduces confusion and waste, and ultimately drives down the time and effort requirements and also the cost. So, and as Andrew was alluding to earlier on, the drop in the number of particular clinical trials sector areas kind of reflect the lack of discordance, and ultimately the fact that that discordance and that ambiguity around what starting materials are at the beginning actually feeds into a reticence to invest more money into an area than later on. So, we all have a collective responsibility to think about these kind of elements in order to realize the value of standardization and opting into consensus standards in order to make the end goal more achievable. So the key thing is that, when we consider iPSC therapy development, it involves many, many different stakeholders. Now we've talked about regulators, investors, ethicists, even politicians, hospitals, small to medium enterprises, clinicians, manufacturers, academia, and patients. And ultimately, there has to be, I suppose, a common understanding in order to efficiently translate to the clinic. And the key thing, in front and foremost, is patient safety. And patient safety can only be, guaranteed or, at least managed if there is a razor-sharp focus on quality all throughout the process of making an iPSC therapy. So the key thing then would be, well, what do we mean by iPSC quality? And depending on who you are, you might have a different understanding of what constitutes quality. So what I'm going to present here is just a brief summary of the organization I work for, what its intentions are, how it's funded, and then basically the activity in the area that we've done over the last 3 years. So as I said, in essence, GATE is focused on assisting the development of standards for allogeneic iPSC therapeutics in particular. So specifically, our mission is to enable the global human community the opportunity to benefit from the new generation of cell therapies by facilitating the development of and access to clinical grade and haplotyped induced pluripotent stem cells for the manufacture of therapy products. So in essence, generating immune matched, good quality iPS cells that manufacturers and developers can use in order to make therapeutics. Okay, so that leads on to discuss the nature of GATE, how it's funded, what its aims are, and basically what kind of stakeholders are involved in GATE's activity. So in essence, GATE is funded by 6 organizations presently. That's the New York Stem Cell Foundation, the Center for Commercialization of Regenerative Medicine, the Cell and Gene Therapy Catapult, the Korean HLA haplotyped iPSC banking initiative, Hong Kong University, and INSERM in France. And what you'll see is from the pie charts on the right-hand side there, is that we're a steadily growing organization which initially contained a large component of academic researchers, but over the last 2 years has been growing further with more industry representation. And also there's clinical staff as well that would be stakeholders contributing to the to Gates' decisions. And the key thing here is that we're developing a holistic view of everything that needs to be done to allow efficient translation to the clinic using clinical grade iPS cells. So when considering the manufacture of iPS cells for therapeutic development, there is clearly many areas where we can help ourselves by supporting standardization. So there's a few listed here. We're going to be talking about quality and quality control guidelines, but there's lots of other areas like consent, clinical acceptability, derivation processes, grading, specification, manufacturing processes, in-process controls, and how we go about getting regulatory acceptance, and basically the formats of key documentation and history files. And with having standard formats, this means that cell lines are more easily shared between labs and jurisdictions and companies. Cost comes down. Ultimately, it's easier for everybody if there's a standard at the beginning. So, like, you can imagine, if you're thinking about like car seat manufacturing, like whether you have a Bentley or a Skoda or whatever, the main components of a car seat are made up of the same components, and they're all made to the same standard, and that keeps the price of the car seat low while still providing the function that is required of it. So in a similar way, by standardizing here, we just reduce the workload without any depletion of a competitive or leadership position, and everybody benefits from it. When I began working at Gate, one of the first things that we did was we canvassed our membership extensively about what constituted iPS cell quality in their minds, how they tested for it. Did they undertake their tests according to a standard operating procedure? Was it done in-house? Was it done externally? And really what we found was a very wide-ranging set of answers with many different ideas about what should constitute iPS cell quality, how it should be tested for. And really it highlighted the fact that we needed to build a visible consensus which would help align the field more. Because when you're moving to a manufacturing stance, really invisible, invalid assumptions, like thinking that your testing is the same as others without actually testing it, can really derail and waste a lot of money and delay a translation process unnecessarily. So basically what this slide really just summarizes is that there was a very wide range of opinions about what constituted quality for iPS cells for therapeutic development. And then through the workshops, we developed a consensus, and the consensus is now published. And basically, we will come back to the guidelines and revise them, as I said. But all this material, including audio from the quality workshops, is all available on the GATE website, listed there. So what the consensus stated was that there were clearly certain critical quality attributes that were mandatory, so-called release criteria, and these would be things like identity, sterility, genetic stability, viability, various characterizations. So, for example, pluripotent stem cell associated marker expression of a certain level, and also your functional assays looking for things like embryoid body formation and directed differentiation into the 3 lineages. And there was obviously, when you're moving from a basic research mindset where the more information, the better, when you move to a more engineering, or if you're going to be presenting the data to a regulator type of mindset, you want to provide information to the regulator where the insights, from that data is very clear. So, for example, you might want to do a whole genome sequencing assessment of your iPSC in the lab under basic conditions in order to have well-characterized, donor material or the iPSC. But then if you presented that, all of that, genetic data to a regulator, they'd say, well, what does this mean? You're showing lots of data here, and I don't understand what this means in relation to patient safety. So really the mindset when you move into a manufacturing stroke gaining regulatory approval mindset means that you're going to make some information mandatory, the release criteria that we furnished earlier, and then other additional information can be collected but is not mandatory. So what can be provided but is not mandatory is listed here. So various genetic fidelity and stability testing data, viability data, other forms of characterization work, and also potency work. So, in essence, all of this is outlined in detail in the publication. So if you want to really get into the nuts and bolts of what constitutes a clinical grade iPSC, the information is there. So that consensus means, okay, we at least collectively within the GATE community know what we mean by quality with regard to our iPSC lines. But then the next question was, well, okay, we know what quality, what means now, but do we test for it in an equivalent manner? And over the last year and a half to 2 years, we've been involved in a very large, intensive process, which is in essence the world's inaugural iPSC quality assessment trial. So we've been doing this in collaboration with the National Institute for Biological Standards and Controls and STEMCELL Technologies, and also CIRA in Japan, the Center for iPSC Research and Application. And we basically undertook a process where we shipped blinded iPSC quality standards to 18 different sites worldwide. And these would include, large, biotech companies, all the way down to academic institutions that have a strong focus on clinical translation. And as you can see there, there's a wide demographic, both in terms of, the organizations, but also where they're located worldwide. So, we're talking about large biotechnology companies all the way down to academic centers in countries throughout Europe, North America, South America, and Asia and Australia. And so that constitutes 18 different sites in 11 countries. And so they were all shipped these blinded standard materials and asked to test the quality of those materials. And we'll be publishing that work later this year. But in essence, just to summarize, we found a lot of learnings, not only from a logistical standpoint, but also in terms of what would be required to generate robust standards, and also what is required to allow participants to have a strong confidence that their quality testing is equivalent to other, quality testing that's ongoing with others in the GATE network. So the key as well is, that the common understanding, we try and spread that as widely as possible outside the GATE community by ultimately by collaborating with other organizations. So we co-hosted a meeting last year in LA, which focused on pluripotent stem cell manufacturing. And really what it was trying to be a new type of meeting where you were bringing stakeholders that wouldn't normally be present when a discussion on iPSC use is present. So, if you go to ISSCR, for example, you get predominantly academic proof of concept type researchers, and you might need, say, a clinical immunologist in the room, or a diehard cell manufacturer in the room, in order to get a full discussion going as to all the parameters that are important. So that's what this meeting was. This was the meeting that we had earlier, last year. And one of the questions was, well, thinking with the end in mind, i.e., safe and efficacious treatments, what kind of iPS cell do we want? And there's a list of some of the elements there that cropped up in the meeting, and these are discussed in detail in the audio and slides accompanying the meeting that are freely available on the GATE website and the other co-hosting websites. So in essence, this meeting was done with ISCT, ISCBI, HESI, CT Tracks, and also CERM. So, the key thing is, each of these organizations are fighting for the same thing, which is to maximize clinical utility of iPS cell therapeutics. So efficient development of these therapeutics, really, as I say, it depends on early adoption of standards and processes, and really, building a holistic understanding as to what's required for the whole process, in order to get to the clinic. So, for example, usability, or the end product that you have, if it needs to be, it can only be administered to patients by highly specialized staff, for example. That could be a big problem in reducing your clinical impact of your therapeutic. So, you might think at the beginning about the format of the therapeutic that you're developing, so that it could be, more widely administered to patients in a greater number of hospitals, for example. So, as I say, in the absence of prescriptive regulation, the community still must seek to align itself. And as we saw with other cell types, the clock is ticking. And as the field develops and the number of iPS cell clinical trials increases, it's becoming more and more important to get common standards and working to key specifications reflecting the end purpose. So in order to develop a holistic view of translating to the clinic, GATE has a series of workshops that again are free for anybody listening to this webinar to access. These deal with many issues, many complex issues, for example, quality control elements, but also, the importance of donor consent. So getting donor consent for a research grade line and a clinical grade line are quite different. And obviously the medical information, the depth of medical information needed to be a donor to be, the source material for what will ultimately be an iPS cell line to be used in therapeutics is a lot deeper than, say, the medical information that would be needed to make an iPS cell line for research purposes, for example. Also, an element that GATE's proud of is engaging with EBISC and Andreas Kurtz in Charity in Germany to develop a database for clinical grade iPS cell lines to be housed, because as we can see from time to time, it can be difficult to understand when clinical grade means different things to different people. You know, what lines fulfill all the criteria that GATE has deemed appropriate for its network, and you know, clinical grade lines that fit the specifications for inclusion in the network can be housed and easily searched from one database. So other elements include understanding the needs of regulators and how to engage them efficiently. I think many of us who haven't had haven't had much interaction with regulators tend to see them as gatekeepers, but really they're repositories of very, very deep knowledge, very broad knowledge to show how things can go wrong when developing therapeutics. And again, you know, we don't want to make the mistakes of the past repeatedly. So, you know, they're there for a very, very important reason, because obviously, if we do things that might seem plausible or, you know, even advantageous to us, but that actually put the patient at a higher level of risk, then that's obviously something that we need to you know, mitigate. It's important that we translate to the clinic safely and effectively, and that can only be done with the wisdom of the regulators. And you know, the point is we need to engage with them in order to understand what they require, but also we need to share information with them, because you know, we know our processes better than anyone else. So there is a dialogue that has to happen. And that workshop there, you know, tells you about how to engage. You know, it's very important when you're engaging the regulator to give a very clear context about what you're talking about, a detailed process. You know, they won't come back to you and respond to general questions which don't have a context, because you know, obviously the answer that they have to furnish has to be context specific. Also, we've workshops coming out shortly that also deal with developing good quality SOPs, so that you know, transition from academic centers to developers is seamless and efficient. And we'll also be dealing later in the year with the subject of licensing and IP. And obviously, licensing for a research grade line would be significantly different from the licensing for a clinical grade iPSC line that'll ultimately might be commercially, you know, might be the foundation of a commercial therapeutic. So with that said, any more information on GATE is available on the website, but we're now going to transition into more of an overview of current clinical iPSC trials, and maybe a little discussion as to where the future of the field is now going. This graph shows the number of registrations made on clinicaltrials.gov. Now, it is important to do your own due diligence in terms of like these numbers. Lee Turner, Doug Sipp, and others have highlighted the importance of not relying on these numbers, because in some cases you might get a listing of a clinical trial, but it would be cancelled, or it might be not up to a specific standard. So really these numbers, you know, they're placed up on clinicaltrials.gov, but you have to do your own due diligence. So at GATE, at the moment, what we're looking at here is at least 12 iPSC clinical trials, and we would be looking at global data and clinicaltrials.gov. One of these has been completed, 4 of these are planned, 3 of these are ongoing, and 4 of these either been withdrawn, suspended, or terminated. There are 3 of these trials dealing with macular degeneration, 2 at NIH and 1 at Kobe Hospital in Japan, 3 for Parkinson's, again in Kyoto Hospital, and 1 in Scripps. 2 which center on the iPSC being used to generate therapies for graft versus host, and that is, you know, with the company Sonata. Another clinical trial which seeks to address thalassemia is ongoing in Guangzhou Hospital in China. And there is other trials dealing with diabetic retinopathy, cardiovascular disorder. There are other trials then that deal with ophthalmological disorder. So 4 active trials are in phase 1, 3 are in phase 2, and 1 are planned for phase 2 or phase 3. So really the kind of countries that really stand out with the iPSC field would be kind of Japan, China, America, and Australia at the moment. So, you know, the current leaders would be Kyoto University and Sonata, in my personal opinion. But you know, obviously these things can change quite rapidly. So in essence, what we're seeing is the number of clinical trials are increasing, and you know, similarly, you know, with the other sectors or other stem cell types or stromal cell types, you know, the key thing is that we should be doing research and undertaking clinical trials in a manner that allows the end goal, i.e., the maximization of clinical impact, to be as likely as possible. So the key element would be that we would align on standards, our own common standards, as a priority to stop history repeating itself to a degree. So if we're just moving on to the kind of conclusions from the webinar, key thing is that, you know, the take-home messages would be that there are invisible, invalid assumptions, and these can easily derail a banking drive or a manufacturing drive to the clinic. You know, by assuming things and not testing them, you know, that's where a manufacturing drive can really go awry. The iPSC field needs to align around consensus standards so it can translate efficiently and maximize clinical impact. Terms can have different meanings depending on who is speaking. So, you know, you might hear sometimes GMP or clinical grade, you know, being used in almost like a marketing context. And it's important you know what the detail around the use of those terms are, I should say. You know, because, you know, the specifications reflect the end use. So it's important you understand the specifications to which things are done, to which reagents are made, in order to verify that they are appropriate for your end goal, so to speak. So, you know, it's very important to paraphrase with people you engage with to ensure that you're understanding the same things using, you know, the same terms, because different professional silos, words can mean different things. And in many cases, you'd be at stem cell conferences where people are using the words in slightly different ways, which then gives rise to huge confusion. Language should be as precise and as accurate as possible in scientific discourse. But obviously, you know, business development and other elements, you know, they'll be trying to attract people to buy products and infer value. So it really is up to you, because you know your process, you know what you want to do your due diligence and ensure all the elements are going to be in place so that you can get regulatory approval for the therapeutic you end up developing. So you need to begin with the end in mind and really do your due diligence all the way through the process. We need to know where the limits of our expertise are and refrain from extrapolating into other professional silos, because, you know, we do have bounded rationality, and, you know, our expertise sometimes would lead us to make assumptions that actually aren't true. So, you know, coming from maybe a basic research or even a developing background, you mightn't necessarily know everything that a regulator needs to know in order to get, you know, approval from them. Collaboration with others, you know, functions, other professional functions, is really key to translating to the clinic. And we really need to remember that, you know, countries and professional silos need to collaborate in order to effectively move iPSCs to the clinic. The ultimate metric of success is safe and efficacious treatment of patients. which means quality has to be integrated into the process from the very beginning. So the remaining conclusions: before initiating a clinical banking or manufacturing effort with iPSCs, you need to understand the regulatory requirements. And as I said, ISCBI and GATE, alongside other organizations, are good starting places. Fitness of guidelines ultimately depends on the end use. So you need to have that clarified and make sure that, do your own due diligence in order to understand what's required. If we wait for regulators to tell us what to do prescriptively, then there'll be a lack of alignment in the sector and we'll see history repeating itself. So it is important that we align. If everyone does things independently, where costs and other elements are going to be prohibitive, it's important that we differentiate between where our process is unique and offers a distinct advantage over other elements of the process that really don't provide any additional benefit. Standards are important not only for research reproducibility, but also patient safety and overall cost of the therapeutic. There are clearly many areas to standardize, but standards are only meaningful if accepted by many. And in the absence of regulations and pharmacopeal guidance, we need to build the trust and the consensus so that the iPSC community aligns. The range of resources available specifically for allogeneic iPSC therapy development on the GATE website, and also would recommend the other webinars referenced earlier in this webinar, particularly David Turner and Lynn Stacey's talks on immune matching and quality in particular. So thank you very much.
All right, thanks very much, Stephen and Andrew, for that presentation. So before we go into the live Q&A, Stephen, I was hoping we could go through a couple of questions specifically about GATE.
Sure.
So how do I go about joining GATE?
Well, if you go to the GATE website at gate.global and scroll down to the bottom of the homepage, there's basically a box where you can put in your email, and then we would send the details out to you. It's free to join GATE, and so basically either email us directly or just put your email in the membership box at the bottom of the homepage.
Awesome, thanks, Stephen. And the next question is, what are GATE's ongoing and future projects?
Well, as referenced, the key thing at the moment is the quality assessment trial, and writing that up and getting it out there. There's going to be a number of publications. One is to do with the logistics of holding such a trial, and the second then is the amalgamated data sets that have been generated and the insights from those amalgamated data sets that show some of the key issues we face for quality testing across different sites with regards to the iPS cells.
Thanks so much. So the next couple questions are more general to the whole presentation. What is the most important criterion to guarantee a good quality iPSC culture or cell line?
So I think there's a couple of ways of answering this question. One is to say that the true test for determining a pluripotent stem cell is to measure pluripotency, which you can do by using teratomer assay or in vitro differentiation. But there's a number of tests that we went over earlier in the presentation that are important for ensuring consistency from cell line to cell line. Things like identity testing is important, monitoring genetic stability, characterizing PSC marker expression, that kind of thing. So unfortunately, I don't think there is one test that's going to ensure quality. There's a panel of tests, but I guess I'd just recommend that you choose your investments into testing wisely, and make sure that the QC you're going to carry out is suitable for your specific application.
Thanks, Andrew. Stephen, did you have anything you wanted to add to that?
Yeah, if you're talking about clinical grade iPS cells, I mean, it's kind of everything that we talked about during this webinar, and they're non-negotiable, because ultimately, in order to get regulatory approval for use of a therapeutic, you need iPS cells to fulfill the criteria mentioned. So the key thing, again, just is like the specification is there to fit the end use of the cells. So, you know, obviously the quality testing is more extensive when you're using the cells as a source for developing a therapeutic that will then be injected into patients.
Great, thank you. And the next question is, do you think allogeneic iPSCs could be better than ESCs for cell therapies?
A few years back, it was, you know, human embryonic cells versus human-induced pluripotent stem cells, and ultimately, you know, you can see there are clear differences where, in particular contexts, one cell type would have an advantage over another. But as scientists, you know, we have to follow the data. So, and I think out of deference to patients, we need to try all avenues open to us in order to tackle, you know, degenerative diseases and injury. So, you know, there are different considerations depending on whether the therapy is autologous or allogeneic. I mean, obviously, autologous cells, you know, taking them from the patient that you're going to subsequently be treating. So there's probably reduced immunological considerations, but then at the same time, if you're moving to allogeneic, you know, if you want to make an off-the-shelf therapeutic that, you know, you could treat large numbers of cells. So, you know, allogeneic allows a large number of patients to be treated, but I think from a proof of concept point of view, you know, probably autologous will give initially the earliest clinical readouts of efficacy, but then when we move to transpose that into a large group of patients, we need to consider allogeneic.
Thanks, Stephen. So just one more question while we have both Stephen and Andrew here. Can you speak to how do I develop quality control assays for characterization assays and potency assays?
Yeah, so as I mentioned, potency is, you know, traditionally measured by the teratomer assay, but this has a few issues. You know, it takes a long time, it's quite expensive, results are quite variable from lab to lab, so it's difficult to interpret the output of that assay. At STEMCELL, we actually use the STEMDiff trilineage differentiation kit, and that's a directed differentiation method. But yeah, the International Stem Cell Initiative actually confirmed in their 2018 Nature Communications paper that in vitro differentiation is sufficient to assess this potential of PSCs. But the only thing that it can't do is measure anything around the malignant potential of the cells. So if that's something you're looking to capture, in vitro diff can't do that. I don't know, Stephen, do you have any input on how people develop QC assays for clinical grade iPSC lines there?
No, I would say that, just to reiterate, that for quality testing, you know, the specification, it really you know, has to reflect the end use of the cells. So what might be useful for determining quality of a research tool might not be appropriate for cells that will ultimately be the source of other cells to be injected into a patient. So the specification has to fit the use. And, you know, quality tests undertaken also need to reflect the use of the cells ultimately.
All right, thanks so much, Stephen, for that presentation there and the questions. I'm just going to continue on now with the live Q&A with Andrew. So just a reminder for the audience, if you have any questions, you can use the chat box on the right and we'll read them out to answer. We just have a few more minutes, so I'm going to try to squeeze as many as I can in. So the first question, Andrew, when referring to the definitions of media, what's the difference between animal component free and animal origin free?
Yeah, good question. So as I mentioned in the webinar, the definitions do vary from place to place, and there isn't actually a
an industry standard. But at STEMCELL, we define animal component free as literally that. It means that the finished product doesn't contain any ingredients that are animal, and that includes humans as well. Animal origin free refers to the product not having been manufactured using any raw materials from animals. So that's the difference. So for animal component free media, that could actually contain like recombinant animal proteins made in animal cell lines, for example. But for animal origin-free media, it's a level higher, where no animal material can be used to manufacture the raw material ingredients. So as I mentioned, this is why it's so important to check the definitions from the supplier that you're purchasing your ancillary materials from, because they do differ, and you just need to make sure that the definition is fit for your purpose.
Great, thank you. So the next question is, how do I find out about the quality workshops and future events?
Yeah, so there's a number of different workshops by different groups. Some we mentioned in the webinar. So Stephen is the representative from GATE, and I'd encourage you to visit their website for info on their future events specifically. I also mentioned the International Stem Cell Initiative and the International Stem Cell Banking Initiative. So this is headed up by Glyn Stacey in the UK, Andreas Kurt at Charité in Germany, and also Rosie Asasi at the University of Miami. And they usually host their workshops together around the time of ISSCR, so about June time. So again, you should visit their website and get in touch with them if you'd like to learn more from them as well.
Great. And so you talked about research grade quality control standards. Are there standards for publishing that should be followed?
Yeah, so again, this varies from journal to journal, and also maybe even reviewer to reviewer. So there isn't really one set of rules on what you have to do in order to publish using PSCs, but what it comes down to really at the end of the day is just making sure you're providing enough evidence to the reviewer that your PSCs are of good quality, and that no quality effects could be leading to some of the results in your paper. So I would say that at a minimum there should be a G-band, and that would be on your starting cells as well as your finishing cells, as well as a pluripotency test. But I think that the more QC you can do, and the more evidence that you can provide, the more convincing the data is. So you should always have that in the back of your mind as you're working. And I know that certain journals are actually looking into stepping up the requirements for publishing. So this may happen at some point, but if it does, I'm sure the journals will, clearly set these guidelines out.
Thanks, Andrew. So I just have time for one more question here. Is there a different recommendation for G-banded karyotyping for clinical grade versus research grade?
So following the meetings that were organized by GATE that were mentioned in the webinar to agree upon the consensus for clinical grade IPS lines, I believe it was agreed that the genetic fidelity and stability should be confirmed by a G-banded karyotype, and the result should be that the line is diploid. So the additional tests like the SNP arrays and whole genome sequencing and that kind of thing, they can be done, but this was classed as for information only. So basically what that means is that data would be included in the package that's attributed to that cell line to help provide as much information as possible, but it's not essential data.